Dear students, dear colleagues and friends, welcome. It is my great pleasure to introduce today the sixth Duve Lecture. As you know, Christian de Duve was the last Belgian laureate of the Nobel Prize in Physiology and Medicine that he obtained in 1974 for his discovery of lysosomes, together with Albert Claude and Georges Palade. The very same year, in 1974, he created an Institute of Biomedical Research, which he named the International Institute of Cellular and Molecular Pathology, ICP in short. The institute, which is located on this campus, has now been renamed after his name, the Duve Institute. And it hosts today more than 320 students organized in 31 research groups. Christian de Duve passed away in 2013, and we then decided to launch a series of prestigious lectures named the Duve Lectures, delivered by prominent scientists from all over the world, talking about their research topics, preferably in connection with the science of Christian de Duve. And that was not too difficult, because the discovery of lysosome that de Duve made, the lysosome plays such an important role in all aspects of cell biology that there are many fields and topics and research areas that have emerged from this discovery. For example, Christian de Duve coined the name autophagy, which is an entire field by itself and will be covered by the speaker of today. We try to hold these lectures every other year, and as you can see on the slide, in 2013, we hosted Michael Hall from Basel, also a member of our scientific committee, who talked about mTOR. Then we had Andrea Balabio from Naples, who talked about the biogenesis of lysosomes and their link with mTOR. Then we had David Sabatini from Boston, Kali Eldin from Uppsala, and Pascal Cossard from Paris. Today, we have the great honor of hosting Professor Ivan Đikić from Frankfurt, who is going to talk about ubiquitin and autophagy. And I will now leave the floor to Professor Jean-Baptiste Demoulin, who is going to introduce the speaker. Thank you.
Good evening, everyone. It's my great pleasure and honor to introduce Professor Ivan Đikić. Ivan Đikić obtained a medical degree from Zagreb in Croatia and then a PhD in New York in the prestigious laboratory of Joseph Schlesinger, where he worked on tyrosine kinases. Then he moved to the Ludwig Institute for Cancer Research in Uppsala, Sweden, where he started his own group and where we met in 2000. And in Uppsala, Ivan's interests switched to ubiquitination, and more precisely, the ubiquitination of receptor tyrosine kinases, such as the EGF receptor. At that time, ubiquitination was considered as some kind of housekeeping mechanism to recycle protein and to drive the cell cycle. Ivan Đikić is one of the key scientists who actually showed that ubiquitination is much more. It is clearly as important as phosphorylation in the control of signaling pathways and biological processes, including autophagy, and of course, with the link with Christian de Duve. And yesterday he told me that actually the reason why we sleep, why we need to sleep, is because of ubiquitination, right? So Ivan Đikić is now working at the Goethe University in Frankfurt since 2003. He's director of the Institute of Biochemistry 2, and he leads two laboratories. One would obviously not be enough, bridging molecular medicine with biophysics. He was awarded several prestigious prizes, including the Prix Louis Jantet for Medicine, the Krebs Award, and the Young Prize for Medicine, and many others. He has published an incredible number of key scientific papers, more than 300, many of them in prestigious journals. I counted 16 papers in Nature, and many others in Cell, Science, EMBO, and so on. I'd like to finish by underlining the commitment of Ivan to transmit his knowledge and passion for science to the younger generations. And I am convinced that you will feel this passion today during this lecture. So, Ivan, we are extremely grateful that you accepted our invitation to give this lecture. And of course, the floor is yours. Thank you so much.
Well, first of all, many thanks for all of you coming here. It's my great honor and also pressure to perform well tonight in the name of the Duve and his lecture, which is a hallmark of the science here in Brussels and Belgium. And I'm going to refer to his work, but I would like to say that most of the cell biology in Europe today would not be where it is without his an amazing biochemical, EM, and cell biological contribution. So it is with the greatest respect to the contribution of one single man in science. And for this reason, we are here as well celebrating this lecture, but it's also very nice to see that Institute carrying his name keeps up the highest quality science, including the education of young people with whom I had the great pleasure to lunch today and talk about not only science, but life, the future careers, and what is the science and why we need to do science more for our societies. So saying all of this, I would also like to thank Benoit and Jean-Baptiste for beautiful words, both for the lecture and for my own career. We are connected via science, but we also connected via the Ludwig Institute, where we all carry the part of our careers, and much longer for Benoit here. And I still remember the days when Jean-Baptiste was a postdoc in Sweden, in Uppsala, and I was leading a young group, as a young group leader. It was amazing how the postdocs have actually contributed to spreading the knowledge also to the group leaders. And this is something which I carry on in a future career, that communication in science is tremendous. And this lecture also shows with your interest that why science brings the people, and I hope you're going to learn a lot. So I'm going to talk about guardians of our life, of our health in our cells. And these are the two principal mechanisms. One is ubiquitin proteasome system, UPS, and second one is autophagy. And in general, most of the studies on them was separate. People studied only ubiquitin proteasome, and other people studied autophagy because they were very complex. But my lab has been lucky to be linked by ubiquitin, which connects both of them. And the last 15 years, I'm not studying them in isolation, but rather, how do they coordinate around the same stress inside the cell? How do they coordinate and keep our cells healthy, clean from danger, and clean from stress? And today, I would like to say that both of these pathways have gotten a Nobel Prize, but the Nobel Prize that actually initiated all of this was the 1974 Nobel Prize to the Duve for discovery of lysosomes, which is the place where most of these large parts of the cell, whether it's a protein, mitochondria, whether it's pathogen, it gets surrounded by the membranes and gets delivered to the lysosome where degraded. In other way around, ubiquitin proteasome system gets the proteins and delivers them to the proteasome. So Christian has not only described the lysosome and peroxisome as a separate, morphologically described organelles, but he has actually put the vision in how the interior of the cell is structured and functionally connected, determining the names of autophagy, endocytosis, exocytosis, and degradation as a principal mechanism of removal of a danger. So just a few words on ubiquitin. ubiquitin is incredible protein. I mean, looks here like a football ball. No wonder we like football. But it is 76 amino acids large. It is incorporated at 5 locations in our genome, such that cannot happen ever that there will be knockdown of ubiquitin or mutation in ubiquitin that will cause the disease. So evolution has secured absolute need of this protein. And then you wonder why this protein can do so many functions. And one way that was discovered early on is that via a set of the enzymes, ubiquitin is added in conjugated manner toward the other proteins, thousands of them. And that can also be reversibly removed by deubiquitinating enzymes. And the number of these enzymes, a number of that, it goes to around 1,000 genes, out of 20,000 genes inside our genome. So imagine, I mean, this is incredible. 5% of the entire genome is dedicated only to modify another protein to another protein. So that gives you a thought, a big thought for quite a long time. And then, of course, the pioneers in the field, Aaron, Avram, and the colleagues, they have shown that ubiquitination of proteins is recognized by the proteasome. It's unfolded protein, and then protein can come inside the chamber, and I beautifully had a discussion today about multiple types of proteasome, immunoproteasome, conventional, large, non-conventional. What the consequence is that you get small peptide, the protein is completely degraded, nothing to be used, but we recycle amino acids, we recycle peptides, or we use these peptides on HLA class and present them to our immune system to recognize us against the foreign, and that's the way how our immune system eventually operates. So that's about ubiquitin, what we knew, let's say, 10, 15 years ago, 20. The second pathway is autophagy, and this is much more broader and bigger. The fact is that somehow cells can form these aggressive membranes which can eat mitochondria, pathogens, remove anything, and help the cell to actually replenish the sources of energy and amino acids and lipids from the lysosome when they get degraded. The issue here is, how does these aggressive membranes know to eat this mitochondria but not this one? And this is the beautiful work by Osumi's lab, who has used yeast genetics to demonstrate that there is a family of genes that decorate the lipids here. These are ATG8s. When they are decorated on the surface, these proteins, together with receptors on the mitochondria, form the bridge, and then mitochondria is degraded, only when this communication happens. And that's called selective autophagy. So in selective autophagy, you can remove mitochondria, ER, pathogens, aggregates, peroxisome, everything that Christian de Duve discovered. So now autophagy has actually helped not only how they are biogenically formed, but how they are removed. And that was a big discovery, and I think the Osumi credited really a great deal of contribution, again, coming from a pure yeast genetics. He never used anything else but yeast genetics to trace the entire pathway. So now, when you see this, you say immediately a question. How can we use these systems to protect us against diseases? And that's one of the big challenges in the last decade. Many of the academics and commercial partners want to do this. And the answer is it's not easy. Particularly, it's not easy for autophagy. For ubiquitin, we manage quite much more. And today, we have several drugs which are in clinical use, which are utilizing ubiquitin system to degrade oncogenes. And probably the best example is one of the worst examples in medicine, thalidomide. Thalidomide was the disaster in the 60s with so many malformations with kids because it was a sickness pill for women who are pregnant. And they didn't study at that time embryogenesis, so they didn't know that in embryonic development, transcription factors can be degraded by thalidomide and cause the embryonic defect. However, in us, adults, these transcription factors do not exist. So thalidomide doesn't do any harm to us. But multiple myeloma is actually dependent on a similar type of transcription factor that thalidomide kills very efficiently. And this is the so-called molecular glue, where thalidomide binds to the Icarus factor, brings the cerebron as a ligase and degrades them, and this is a multi-billion-dollar drug used per year. Incredibly efficient. And you can see how little prediction here it is, how crisis leads to the success. And today, the scientists like Craig Cruz and Ray Deshay has evolved from this small molecular glue to a much more Lego kind box where you have a small chemical binder to ligase, and you have a small chemical binder to the target that causes the disease, and then you have a linker between them so you can synthetically develop new drugs which are going to degrade estrogen receptor, androgen receptor in prostate versus breast cancer, and many others. More than 30 different clinical trials are currently ongoing by utilizing the power of ubiquitin in a very systematic and pharma-based drug discovery. So in order to address this issue, we have also developed in Frankfurt a network or cluster which is called proxy drugs. And there we are going further apart. We are not only looking for degradation, but you can imagine that you can take the chemistry and bring two hands together to do something else, to regulate transcription, to bring the protein to the membrane or to the cytosol, and remove the, for example, BCR-able translocations and so on. You can actually do many, many more things, and this is what is very active part of the research today, both combining academia and pharma world. I'm not going to go about details about this, but I'll show you how this cluster, because we are running it now for 6 years, in which we have 5 universities, 11 companies, and the money comes from the federal government, $3 million a year per year for 9 years. This amount of money you can do a lot. You can revolutionize your technology platform such that you're faster, more deeper, and eventually much better in scientific output. And I'll come back to the last part of my talk. When we use this platform, in no time we discover a new drug that you will see was important for the cancer treatment. So now coming back to history, we heard about my interest on ubiquitin as regulator of endocytosis. And this we did in Uppsala, Sweden. I moved in Germany, and I had a young student from Poland, Krakow. He came out and said, I would like to do a project. And I gave her a project to ask the question, why and how ubiquitin can function to such an extent, to so many different functions that were alluded, but we didn't know how. And in order to do that, at that time, one of the most modern technology, today's people laugh on this, was yeast-to-hybrid system. And yeast-to-hybrid system was very powerful because what you can do, you can put ubiquitin and put a library to complement each other, and only when they bind, yeast grows. Very simple. But you need to have the right conditions that you don't have false positives. So we did very good yeast-to-hybrid system screened by ubiquitin as a bait, and we come out with 200 genes or proteins that bind to ubiquitin and rescue survival. So we said they must bind together. What is their role? Then using bioinformatics, we identified some kind of different folds as small parts that bind to ubiquitin on many different layers. We solved the structures of each of these domains binding on a different surface of ubiquitin. So ubiquitin is not a ball. It is actually globular structure with multiple domains which can interact with other proteins, therefore giving them not a single type of signaling, but multiple intracellular signal that can communicate now with many different pathways. And indeed, I will show you today just the 2 which were published, and the third which took us 20 years to understand. So the first paper that came out from Magda's work was absolute breakthrough, because in this paper, she showed without any doubt that ubiquitin does not need to be connected to proteasome to fully function medically and biologically. What she did, she was focusing on the UV-induced damages in our cells, which happens all the time when we are outside. The marker of damage in our cells is biochemical sign of ubiquitin being added to the pCNA. And pCNA is the trimer which surrounds the DNA. So when you have this, you know there is a damage. And this damage needs to be resolved. In order to resolve this damage, you need to recruit this enzyme to the ubiquitin. And the way how it's recruited, it's via this ubiquitin binding zinc finger that Magda identified in yeast-to-hybrid system. And then she was able to show when you do not have this binding, you there is a human disease, inherited disease called xeroderma pigmentosum, where you have multiple tumors everywhere where the UV light hits the body, but not where it's not. So the only therapy is no UV light, which is difficult to live. And what you see inside the cells, you see that the normal cells have all of these foci, as happened here. They are very nicely recruited. When there is no ubiquitin binding function, there is no foci, therefore the cells are damaged and the genome is damaged, causing the tumor development. And that was really pure. There was only recruitment as the function of ubiquitin. There was single ubiquitin there. The second protein that came out from the screen, which was very exciting, was named NEMO. And the NEMO is the actually protein that binds to, or is required to TNF signaling, cytokine signaling, to survival, gene regulation. This comes in, and we didn't know what to do with this because there was no binding to single ubiquitin. Only when we solved the structure, we recognized that NEMO binds specifically to diubiquitin. And the diubiquitin was not normal ubiquitin with lysine linkages, because normally conjugates to lysine. Here we had methionine to the end of the other one, so it's linear ubiquitin. And we solved this, and we were very convinced that this is right. Of course, there were a lot of resistance at that time, but Kazuo Iwai's lab has also shown that this is important in living cells. But why we were convinced that this is right? Because it was the first structure of a diubiquitin with the domain, and in this case was only alpha helix. It's not a specific domain. But the amino acids here have been mutated in ectodermal dysplasia kids with immunodeficiency. And these mutations actually mapped exactly here, here, and there. So you had several mutations targeting the site of binding between the NEMO and the linear diubiquitin, proving indirectly functional importance. And of course, there was many issues there. We proposed that there was NEMO binding to linear ubiquitin, and this is done by this ligase, and that leads to the NF-kappa-B activation. In order to prove that, another postdoc in the lab, Fumio Ikeda, proven by using mouse models, when you remove the ligase, there is no linear ubiquitin, no NF-kappa-B, and you have cell death as a consequence, particularly in the chronic dermatitis of the skin of the mice. So these were the two, again, nothing to do with proteasome degradation. Here is an absolute signaling tool that NEMO binds to the linear ubiquitin, which is created by another ligase bound to the TNF receptor. And that's essential for cytokine signaling. So now the third one, protein that we focused on out of these 200, there were many other publications, but this one was very exciting. This one was exciting because it also has UBZ domain, but what it had the catalytic protease domain, metalloprotease, Spartan-like. And had also the other characteristic binding PCNA, P97. Everybody got excited. This is another UV responsive gene. Even though several papers came out, we knew it is not UV responsive. Because I was contacted by a human geneticist from Ulm one of the evenings after 7 o'clock. I said, who is calling at 7 o'clock from hospital? That must be important. And the guy tells me, we identified mutations in the gene that you described in your first paper that contains UBZ domain, but we didn't know what does it do. And the mutations in this gene comes out in the family of Moroccan family, where the child had very obvious signs of aging. You had kyphosis, you had eye problems, and it was all driven by the genome instability and the stress on the replication in those kids. And what was very surprising, it died at the age of 16 due to the hepatocarcinoma, aggressive hepatocarcinoma, no viruses, never detected in a human population with such aggressiveness. And the mutations in this child were point mutation here in the tyrosine 117, which is hypomorphic mutation. It's not full null. Because if you would knock out it, all the mouse die by knockout of Spartan. So it's absolutely essential at day 3 of survival of life. So as important. So this was hypomorphic, and the second mutation came out from the C-terminal tail deletion, also hypomorphic because it can come to the nucleus not completely, but partially. And there was then, when we were publishing this, we went to the meeting, I talked about this, there was another family from Australia having 2 boys. One died at the age of 70, and second one was age 11 at that time. He got liver transplant, and he lives still today. He's 21 years old. So that's what we knew, that this is something with the stress. But we didn't know, again, what is the biochemical basis that this happens in kids. And then this was now a few years afterwards, 4 papers came out and described that spartan is metalloprotease that eats proteins which are coupled to DNA. That means that the DNA binding and ubiquitin binding to these proteins activates their protease activity, and then they cleave everything that is coupled to the DNA. And that was shown in yeast by the late Stefan Jensch's lab. This was in our world, Christian Ramadan's lab, and Simon Bolton and Julian Stingerle showing. All of us showed the same in a different settings. So we knew that the problem with the kids is accumulation of DNA protein cross-linking, which is incredibly frequent, but not so well-studied process in a DNA replication and repair. And indeed, if you look now theoretically, DPCs are done either by external signal or via enzymes. So external signals can be formaldehyde, which is most commonly. That's the reason kids die at very early on. And that's the reason when the mother drinks during the pregnancy, which is teratogenic, because alcohol gets converted to aldehyde, aldehyde causes DPCs, DPCs damages embryo. So that's the chemical nature, because aldehyde is going to take any histone and couple it immediately to the DNA. And that happens also with irradiation, UV light, different type of chemotherapeutics, and so on. But there are also other drugs like inhibitor of PARP, camptothecin, or etoposide, in which they couple exactly the enzymes they inhibit directly to the DNA. And you can imagine when you have this, or this, or this, the replication cannot go further. So it's replication stress, replication fork is blocked, and no further promotion. So in order to remove this, we have developed through evolution pathways to block that. And major pathway is the spartan or WSS1 in yeast, but there are several others. But the spartan seems to be the most important and the most powerful. So now we came out to know what is happening. We came out to know what is happening with the human genetics. We wanted to understand why is children going through progeria, and why do they have a liver cancer. So in order to address this question, we came with the 2 projects. One was the young medical doctor from Croatia who was working 2 years in emergency room and got completely disillusioned by the system at home and said she wants to do PhD. And she calls me, completely not unknown, just writes an email and calls, she wants to do PhD. And I accepted her, and I actually didn't know whom I am accepting. I just knew that she's very motivated. She did amazing work with this, and you will see all the results. So out of no training before, he now finished the PhD. She got the medical degree accepted in Germany, and she's now doing the dermatology specialization. Incredibly driven lady. The second experience is the veterinarian from Spain who came in the lab as a postdoc to study ubiquitin system in cancer. And he asked the question, what happens in the nucleus during this stress? So Ines did mice, and mice, here's one experiment, 6 years, 900 mice being crossed. This was one of the most expensive projects that was run in my lab. So what she showed here is that when you have a knock-in, one or deletion here, the survival of mice is much smaller, not 25, but only 8%. Secondly, any mice that is born of this 8%, it's immediately genotyped by vision. You see it, gray, kyphosis, and so on. You don't need to do genotyping. And the third finding is that most of the female mouse were significantly worse, and there were less of them born. So initially we thought that the mutation in kids was specific to boys, because only boys were born. With mouse, we feel that it's actually much more damaging to females because they all die, and only survivors who are less damaged are boys. And we believe it is because testosterone is protective, while estrogen is harming to inflammation. And I'll come back to explanation of that later. So now, in the same time, Christian comes on and does mass spec and shows that the spartan doesn't bind only to S phase, which was published, and we knew that from 2016. but it also binds to many mitotic proteins. And it is also triggered in the very early prophase of mitosis, expression of spartan. So all of this was important because when we remove the spartan from cells, this is when you see here the normal the mitotic cycle. You can see that when something happens in the wild type at 40 minutes, it is very much delayed in the lack of the spartan to 95 minutes. And when you look at the end of the results, almost 60% of cells that have defects in mitosis, whether it is catastrophe, here total catastrophe, or you have here the micronuclei formed next to the nucleus, or you have this spindle which are connected multiple spindles. So all of these three events, when you do not have a spartan, you have high level of DPCs, you have all these damages in mitosis. And that was important because the consequence of defect triggers on inflammation in the cytosol. And we discovered that by looking for both in the cells, you again have micro, and you have also cytosolic DNA. Here is the pico green, and you can see a lot of DNA leaked in the cytosol where there is no place for it. And you can see the same one in the primary tissue of liver. Here you can see cytosolic DNA coming out, and you have micronuclei all around, statistically significant. Consequence of this is activation of STING and C-gas pathway, because we know that anytime the DNA comes in or foreign RNA comes inside the cytosol, STING and C-gas will be activated, or can be activated. We proved that very nicely by showing that in the knockout of spartan, you have tons of the phosphorylated STING. You can see here staining, green, as compared to the cells which are not damaged. And you can see biochemically, everything goes up. STAT, STING, TBK1, IRF3, just name it, double-stranded breaks. All of these damages happen as a consequence of the lack of the DPC resolution. And then we show that this all happens, but now when we overexpress the wild type spartan, we rescue all of these phenotypes. They're all rescued. But they're not rescued if you put the mutation from the patient, tyrosine 117. So that really proven biochemically that the defect is coupled to the protease activity of spartan, and the spartan can recover it even when it happens. So then, and the consequence for this is that you had the interferon signaling, and we saw it immediately in the liver, incredibly high signature by RNA-seq or by proteomic analysis. Many of the proteins are innate immunity, response to interferon, they just go high in the liver of the knock-in mutation of the patient. We asked the question whether this is unique to liver, because it might be that the liver is kind of specific and therefore we have liver cancer. The answer is no, it's all over. And the highest level of interferon is in embryo. And therefore we believe that most of the individuals die in the embryonic development due to chronic high activation of interferon STING C-gas pathway. In order to prove that this is so, we now cross the mice. So we now cross the mouse of spartan knock-in, C-gas knockout, spartan knock-in, STING knockout. So if this goes by this pathway, they should rescue the phenotype. And indeed, this is this 8% which survived. Now if you put it to 100%, this is the 8% is 32. This goes almost double to 60, 70% survival. So the life of 50% more mice have been rescued completely. And by using the STING inhibitor, which we worked together with Janos and Andrea, using this inhibitor, we injected in mice. When they gone birth, almost 90% of mice have been rescued of embryonic lethality only. And this doesn't stop with embryonic lethality, it also projects to the progeria. So this paper actually proven that aging starts during the embryonic development with obvious signs of aging, and we can monitor the aging through embryonic development. And then we can monitor it also even through the rest of the life, and we can see that most of the phenotypes, not completely, but partially or strongly, are rescued by the lack of STING and C-gas. So this all really gives us a strong evidence that this pro-inflammatory chronic pathway might be underlying many of the phenotypes with kids. And now the question is whether this also happens with the liver, because the liver cancer was the cause that the boys died. And the answer to that one is yes, in mice, we have also a classical HCC liver carcinoma. You can see it here by histology, by morphology, and 90% of all mice that were born by the age of 75 weeks, they all finish with HCC. Incredible. Not a single other tumor has been identified, while every cell in the body has a chromosome instability. And the answer for this one, we still don't know. We have no understanding yet why it is so specific to the liver, but what we can also say that this mouse model now gives us an incredible power to study that and to translate that to the new potential patients. And while I'm seeing the last year, there were 2 new families, one in Manchester, one in Paris, who identified the other kids, and first time the girls were identified, and they died at month 3, and the 3 years, the sister is still alive. The young one had a complete liver destructed, and the 3 years while all got a liver transplant. So now this mouse will probably give us a chance to maybe use anti-JAK inhibitors, which are downstream of interferon, or potentially when we have STING inhibitor. I'm certain this treatment of these girls through time is going to give them much, much longer life. So I think this is the way how you come out with the findings without wanting it, just following your own science. So I would like to finish this first part by saying that we have identified a pathway that starts with the lack of the function of spartan, accumulates DNA-protein cross-linking, leads to the mitotic distress, formation of micronuclei, activation CGAS, STING, regulation of interferon, and this drives premature aging from the embryogenesis to adulthood. And there is also liver cancer development, which we connect to this one, but we are still not understanding full specificity how this is regulated. And this part of the work has been really driven by Ines and Christian, and has been a long-term collaboration with Janos's lab in Split University in Croatia. And now I get 2 additional postdocs, both from Croatia, so it's quite much of the Croatian project in my own lab. So I'm also proud of the accomplishment of the people from Split. So now I'll switch the gear, because this was a tour for the last 20 years, how we study ubiquitin binding protein. But then when Christian came in the lab, he was interested in deubiquitinasing enzymes, those that remove ubiquitin. And he was interested in a cancer development. So he comes in the lab and he says, I want to work on USP39. It has been shown to be oncogenes in many of the MYC-driven cancers, which are very aggressive cancers, lung, liver, several others. The problem here is that USP39 is catalytically inactive. Biochemically, we've proven that. Second of all, there was identified in yeast that is component of 3-SN-RMP complex, which is part of the splicing. And they indicated the role of oncogene, but nobody knew how. So we were surprised by this finding. We use the cells, knock down USP39, and what we see a lot of ubiquitin, much more ubiquitin as compared to the wild-type cells. And this was illogical, because if you remove DAB, does not have an activity, you would not expect a ubiquitination. You would expect no change in ubiquitination. And by this observation, we went on to analyze why there is an increase in ubiquitination in the absence of the catalytically inactive DAB. We were also able to show that they are forming some kind of ubiquitin aggregates here inside the biochemical assays. So the important part came out when we did EM analysis. We just pulled out USP39 from cells, and we pulled out the entire 1.8 megadalton complex 3-SN-RMP. And by work with Galai's lab from EMBL, we solved the structures, and we were able to show that when there is USP39 here, there is a stable complex of all these 30 subunits. When there is no USP39, the complex gets dissociated. And this was proposed by the fact that in yeast, SAR1, USP39 in mammalian, really needs to be recycled inside the splicing cycle. And this comes out to this slide, because splicing is really megadalton complexes, which actually enables us to cut down introns, connect exons, and make them mature mRNA that provides fidelity of our translation and our life. These complexes are not stable. They actually come and go. And you have the 2 major ones, U1 and U2, which are catalytically sized, which will bind eventually here. And then you have this U4, 5, and 6, 3-SNRP, where the USP39 binds here. This binding is important to come and land on the side of splicing, but then soon after, USP39 and U4 go out, and 5 and 6 stays and do the job. So somehow the role of USP39 was in this cycle here. And the other important part was that several components of these 30 proteins were identified to be mutated in human disease called retinitis pigmentosa, where the people damage their rhodopsins and visionary neurons, and they don't see, particularly during the night initially. And the mutation in USP39, when you do it in zebrafish, you have retinitis pigmentosa phenotype. We could not do it in mice because they are embryonic little. So then we asked the question, what happens in the cells? We did a lot of experiments. And one experiment that became incredibly strange, but also very provocative, was the following. We knocked down the USP39 and then sent the material to the RNA-seq. And we asked the question, what are the new splicing events which are happening there? Is there any defect? And it happens that you had 50% of alternative 5' splicing. Alternative 5' splicing means that the normal splicing from here to there does not land here, but it jumps before or after, alternative to the classical. And we did quite a lot of work to explain that. It was difficult biochemically. Eventually, mathematically, we explained that this is a kinetic model in which stability of the complex selects correct side. If it is unstable, keeps flying left and right without any logic. Anything that will be looking like splice side, they will land there. But what was very surprising is that 70% of all of these events have not been annotated in any of the deposited human genomic data. None. So this tells us that we have 70% of novel potential peptides, novel potential antigens. And of course, when you have this type of clear data, but they are also incredibly provocative. I was suffering to the last day of our publication because you always worry, are we right or are we not right? Maybe this is not as clean. We did it in 2 cell lines, but nevertheless. The 2 weeks before our paper comes out, a year and a half ago, there is another paper coming just 2 hours in the same journal, and this is the paper from Juan Valcarcel from Barcelona, the person who has spent 30 years studying splicing. And his paper was about knocking down all 305 splicing factors, which are in this big box I showed to you, 305. He analyzed all of them and analyzed all splicing defects. Amazing paper. So, of course, I rushed in the paper. I'm looking, where is USP39? And luckily, it lands here. USP39, 3S-RNP complex, very much the alternative 5' splicing. This is one of the strongest 5' splicing defect. And then, even more, he looked here a fraction of novel junctions, novel meaning not known before, comes up the top of it, USP39. So we opened the bottle of champagne that day, pre-2 weeks of our paper, and celebrated their paper and our paper together. Because that really gives you incredible confidence that the data were strong and good. So now, we come to the point to explain the ubiquitin as I started with. Now, when you look, what happens in the cells with these splicing events? They are going through the mRNA control. They are going through the translation control. They're expressed. They are not blocked. What happens with them is that they come out in a cell. Here you have a knockout of USP39, a lot of aggregates, EM, here, and you can see them that they are ubiquitinated. So this ubiquitin signal is not because of deubiquitination. It's because of the more aggregates which are ubiquitinated as a consequence of their aggregation. Now, autophagy and proteasome would like to remove them. And indeed, that's what happens. Not only that in proteasome they are removed, but in the ER, you have an enormous number. These are all red dots, are missplicing, error splicing messages, which are found in the ER. And in the ER, they trigger on all the 3 major stress pathways. So the missplicing proteins, they bring the natural proteins and themselves forms the aggregates in cytosol with proteasome, ER with autophagy, and they are being removed. If they are not removed, eventually cells go to the CHOP-dependent cell death. And indeed, that was published, and we really were happy to see the other paper published, but we were able to show that cryptic splicing in the lack of USP39 leads to alternative 5' missplicing that goes to the prototoxic stress, and that is removed by either ubiquitin proteasome system or autophagy, and that these are two guardians of the damages or the errors in the splicing. So that's all very beautiful. But we asked the question, or Christian asked the question, why is USP39 oncogenic? That was his start of the PhD, of postdoc. It took him 2 years to come at the beginning. So he went on to clinic and look what happens with USP39. So he worked with our pathologist, T3, T4, hepatocarcinoma, an enormous amount of USP39. All of this brown is USP39 as compared to these stages or healthy liver. He also showed that there is a correlation between expression and severity, and he also showed that whenever you have a high level of USP39, patients die much more because they are in aggressive phases, all fitting. But because we had our pathologist, we use now the 3 samples from here and 3 samples from there, and we ask the question, what is the difference inside their cells? And in order to study that, we did first mass spec. These are 3 low, 3 high, ran mass spec, and we see that in the high level of USP39, you have protein folding, ubiquitin proteasome system, ER stress, all the things that we already described in the absence of USP39. But here we don't have an absence. We have actually high level of USP39, and the tumor cells are living under stress because they don't have it enough. But they still live without any major damages because by evolutionary they keep selecting to survive. This was also shown by RNA-seq. You have the same signatures, high level. So this data, together with pathology, tells us that in the malignant cancer cells, you have a high cost of living. This is like a party in Ibiza. You have a living until night, 2 nights, 3 nights. The energy that you do this, cancer cell can tolerate because they have machinery. And they live. In the normal cell, that's absolutely not the case. You have much lower level, but also much lower level of proliferation. So now, if this is correct, you can always propose that these cells are now very sensitive to removal of USP39, or they would be more sensitive than to the healthy cells, for the reasons that I described. They are living high cost. But they are still, as I want to say, they are living still normal. They are not dying because of this. Maybe some, but majority lives normally. So now we said, okay, but let's block then the splicing in these tumors. And we went to literature and said, splicing has been targeted for 25, 30 years in cancer. Many pharma industries have done the same approach because they knew splicing is absolute requirement of many of the cancer cells. And they discovered more than 20 different inhibitors. Some of them went to clinical trials even. And they were all stopped. And they were stopped because all of these inhibitors were targeting the catalytically active site or major site of splicing where the action happens. So because of this, you are blocking all the splicing, even in the cells which need to splice a little, but you block that. So, and therefore they also have during clinical trial, there were some side effects and they were stopped. So now we would like to say is that maybe not blocking this, but blocking that part, or not even blocking it, but introducing the errors. You are actually not shutting down splicing. You are making a splicing being error prone. So in the high level of cancer, by removing USP39, we believe we are going to introduce more and more and more errors, as I showed to you, proteotoxic splicing events, and that can kill these cells much more efficiently. So that was hypothesis. And indeed, in order to prove that one, we went on now to establish the tumor model, which might be done in relatively fast, not to do this work that Ines did for 5 years. So this model is quite good for message, not for reproducing the complete HCC. Here you have hydrodynamic tail vein injection. You take different plasmids and you inject them in the tail under high pressure. These plasmids get completely taken by liver cells inside because they're under pressure and they just get absorbed. And once they are absorbed inside the liver, something happens. And what happens is if you add oncogene like c-MYC, or if you remove p53 tumor suppressor, in 30 days you get hepatocarcinoma, many of them, just showing up. In 100 days, mice are absolutely loaded by tumors, and we need to stop the experiment. And you can see that these tumors are actually high-level MYC, no p53. So exactly what we did. And what was nice to see is that in the same mice now, you have the USP39 going up. So the tumors, while they are developing, they are increasing 3 times level of USP39 because they require this protein to keep up with splicing. So now we ask the question, in these tumors, if we just suck down the USP39 by genetic approach, what happens? So what we did here is, instead of only putting p53 knockdown, we add now overexpression of mouse USP39. And when you do this, you have many tumors in the wild type, but even after 100 days, there are very few tumors appearing, or very small ones, when we remove USP39. So this tells us that all of these tumors died through the crisis of the lack of USP39 and prototoxic stress. And I'm not going to go into details, but we see everything, huge amounts of aggregates in tumors. But the more relevant experiment was the following. Now, instead of mouse USP39, on the top of it, we put the human USP39 because it was insensitive to the shRNA. So now we introduce the human, and we could rescue. Here there are no tumors. You put the human USP39, tumors are growing again like a big party. So that is really genetically top-level proof. And we then be able to conclude that in the high cancer cells, if we genotype them by, or stratify them by USP39 high levels, we can actually create a drug that would remove that, would lead to prototoxic stress, dying of the cancer cells, preferentially. And now you want to block this. This has no catalytic activity. You cannot use inhibitors. You cannot do much. But what you can do now, there are 2 approaches. One is the direct strategy, developing the degrader, PROTAC. And the second one was, which we came out with, that high level of USP39 is driven by the lactate, which is metabolic consequence of the liver cancer aggressiveness. And the lactate via MYC expresses USP39 so high. So if you block that, USP39 is going to stay down. So now I told you that we set up these proxy drugs, and without proxy drugs, there would be absolutely no way that within a year and a half we could develop something that looks drug-like. But together with Xin Lai, who is the group leader in proxy drugs, he took splicing compound library, he took DNA-encoded library, he came out with multiple binding fragments. Out of them, he made PROTAC containing USP39 binder and VHL ligase binder, or USP39 and cerebron. VHL in our hands work much better, and we were able to show that they can degrade USP39 specifically in cells. And we did a lot of other work. It's all published in the very recent Angewandte Chemie. But just for the sake of the final stage, I can show you that liver cancer cells, when you treat them with this PROTAC VHL, you can degrade in a concentration and time-dependent manner USP39 very specifically. And you can do the same one with oxalate, which is inhibitor of lactate, and this is in much, much higher concentration, so this one needs to pay attention a little bit for therapeutics. But what you see in these tumors, moment when you start decreasing USP39, you increase prototoxic stress. You see the proteins being aggregated and accumulated in a cell. So we have a drug treatment, and we have prototoxicity, and cells are dying. So now we went on to do the mouse work. And in order to do mouse work today in Germany, you need to be Seinfeld magician. Because in order to write all the paperwork, it needs you 1 year, 2 years, 3 years. There is no experiment you can do under similar conditions. So instead of this, one can just write an email to China and ask them, can you do that within 6 weeks, or 12 weeks, or 16 weeks? And they say yes, and the price is less than 1/5 of my cost in Germany for writing the license. So that's the reality where we are currently in Europe. Absolutely non-competitive, particularly in the light of this very important experiment, because you cannot move further. So what we did now, we established and sent everything to China, and they did the work. They put vehicle, PROTAC, OXA, and this was the protocol we write for them. They took everything at every stage, 1 week, 1 week, 1 week, looking everything. And we look for the normal mouse kind of Weight, no changes, so there was no toxicity. We look in the organs, liver, kidney, spleen, no changes in any of the treatments. We look histologically, no changes. The only change what has happened was in a tumor. And in a tumor, when you look here, you see here the control. You have quite a lot of USP39, which we know it is grown up in aggressive tumors. We have very high proliferation index. And you have very few cells dying, which is okay, because they normally die under this high cost of living. However, when you treat PROTACs, you see very little of USP39 present, no proliferation, and many, many more apoptotic cells. So that's the dream. You have no tissue of the normal cells, and you have a tumor really going down with proliferation histologically. Similar happens with sodium oxalate. And then when you look for the survival of mice and the tumor volume, after some time we need to stop it, but the normal tumors grow exponentially, and the tumors treated by both treatments stop relatively soon, and they really don't grow significantly. So this experiment gives us a strong evidence that actually we can use this. Even though we didn't study PK and PD of these compounds, we need to work much more on them. But even this experiment tells you that in vivo, in mouse, you just give them in food, they work. So I think this is really great boost for us, because we would like now to follow up on this one, because we believe that high expression of USP39 in aggressive, not only liver, but we also have evidence for lung cancers, can be therapeutically exploited by using the degraders of USP39 as a new therapeutic approach, which is quite powerful these days. So I would like to stop here and leave some time for questions. Again, here Christian's work was incredible. He is now a group leader in Sevilla in France. And there was also amazing work by our collaborators and funding by Proxy, ERC, and DFG. Thank you very much.
Thank you so much, Ivan, for this fantastic lecture, and I'm sure there are a lot of questions for you. Misha?
I can begin with something really nice that goes back to the beginning of your talk about the multiple copies of ubiquitin. That was really fascinating. So typically we think that there would be duplication and then some degree of divergence. So are there amino acid differences between these multiple ubiquitins in places where they interact with some of these other proteins?
Right, not at all. I think there was an evolutionary pressure to keep the normal, same, very highly validated ubiquitin expressed. So there are two genes which are expressed as a chain. So it's not one ubiquitin. It has ubiquitin, 4 or 8. When you express them, you get immediately 8 monoubiquitins because the proteases cleave between them and make them ready to be engaged. The 3 other genes are part of the gene chimera in which ribosome subunits have been fused with ubiquitin at the end, and they are produced. They have ribosome protein and you fuse to ubiquitin. So that's the protein that's produced. And as soon as it gets out from the ribosome, proteases are waiting and cleaving the ubiquitin, and ribosome subunit is then native and can do the function. And there are 3 examples like this. And that is for the purpose that life without ribosome also cannot happen. So you need to express also a ribosome subunit, so you constantly keep expressing more and more ubiquitin. And the amounts of ubiquitin in our cells is predicted biochemically to 20 micromolar, which is very high. So for that reason, ubiquitin is all over, and you don't need to kind of transport them to the sites. It's actually quite frequently at the site of the action.
And is there any evidence for like dose effect? So if you were to get rid of half versus 3/4 versus...
Right. This is a good question. There are some evidences that the ubiquitin is saturated, meaning there is always enough. But there are also evidences when you overexpress ubiquitin, and you particularly overexpress ubiquitin with specific ligases, amount of the impact of this ligase is significantly higher. But this is a condition when you have an enzyme that utilizes the co-expressed ubiquitin in the most efficient manner. It does not represent the fact that at physiological level, or even pathophysiological level with high aggressive tumors, that there is no sufficient ubiquitin for anything the tumor cells wants to do. So I do feel that evolution has created, first of all, enough, and second of all, protection of any of the errors by genetics or by pathogens. And also the other question, which I didn't talk about this, but most of the knowledge we learned about ubiquitin comes from studying the bacteria and viruses. And the fact is that bacteria and viruses do not have ubiquitin at all. But we learned from them because these pathogens have used the genetic transfer and utilize our own enzymes like ligases, DAPs, make them as weapons. They inject them in our cells, and they use our endogenous ubiquitin to do whatever, shut down innate immunity, reorganize organelles, fragment mitochondria, and so on. And this information we learned from bacteria because the ligases were very convenient, because they could be expressed and injected in cells.
So on the last slide you showed so the graph with the tumor size with the control and two of the different drugs. And we can see that for the drug 2, it's starting to increase slowly from day 35. When a way we could say is could get exponential. Could it be interesting to continue the experience after day 49 to see the impact of the drug on the longer term?
Right, this is correct. But even in China, you're not allowed to do that because the control tumors are so large that we also need to be as concerned about animal welfare under these conditions. So this is not done. But you are really good, have you good eyes without glasses? So here what you can see that, if you would just emphasize here, even at the very, very early stage, you already have very strong impact. And this strong impact stays, changes a little bit, and we know that oxalate has a stronger effect because we believe that it has a stronger effect because it also impacts metabolism and lactate and all other things that tumor cells need. So it's not only USP39 in this case. So this effect of this stuff is a bit stronger on that aspect. But clearly, this is a huge difference, and this experiment doesn't need anything else to conclude, but there is a capacity, and now we need to really work hard. We are already applying for the grant to get a small startup company in Germany. They have these several programs that can give you advice how to do that, and they will support you. They'll give you money before you need to go to venture capital, such that you have better conditions when you discuss with the people who has money only in their mind. So you need to be a good negotiator there, so protect your discoveries. And that's what we are doing at the moment, trying to do some maturation of the chemical matters, and also do PK, PD, try to see when it works the best, and what are the conditions when we can give them.
Thank you for that. I had a question back to the first part for the SPRTN mutant. So I was wondering whether the phenotype that you see that seems to be specific to the liver of the cancer, whether it might be related to the fact that most hepatocytes are polyploids, so that's difficult to handle mitotic spindles in those conditions, and might explain the
Right.
liver specific.
I was talking about this project for decades. That's how we got Australian family. Anytime I go somewhere, in 30% of cases, this topic would come out. And we went on to really study that because it is logical. But then when we look for polyploidia, the liver cells are fifth in the line of all other tissues which have polyploidic cells. So this cannot be explained by this phenomena because many other cells have also polyploidic nature. And I mean, at the moment, our hypothesis is that liver cells are very sensitive to inflammatory pathways. We know that because normal liver cancer develops via steatosis, chronic inflammation, eventually adeno and adenocarcinoma development. So that's the primordial sensitivity. But the second part is that I believe it is important, but we need to do it now with conditional knockout. And it is about the aldehydes, because most of the chemistry from guts go to the liver first. And this chemistry is huge. So now imagine if you have any formaldehyde, aldehyde, alcohols, anything that converts to aldehyde, the liver cells are going to be heavily exposed. And this hypothesis was quite good, and we actually work on this more now. One of the problems, and this was also in our paper, is that amount of DPC does not automatically mean you have a major problem with the cell. Because in the brain of mice, and of course we don't know with kids, but with brain of mice, they have the higher level of DPCs than in the liver, much higher. And they are non-proliferative. So the DPC there doesn't have mitosis, and therefore they don't have all this, there's no interferon in brain, but the amount of DPC is there. So we need to now couple all of these type of things and then try to study most likely in a liver-specific manner with conditional, because this all knock-in everywhere. We would like to make now conditional knock-in in liver cells alone, and then try to see whether it's hepatocyte-specific or immune, the Kupfer cells and other system around. But it's a big challenge, and I think it's not going to be fast either.
Thank you. It's the next point, and as expected, I learned a lot. So I just have a question. I'm curious. I was wondering if this USP39 has any preference for some specific splicing site. In other words, if a body will choose to get some beneficial 5' splicing by selectively avoid this USP39 at certain point, at certain place.
Right. Yeah, this is a good question as well. I think we were surprised by how misled we were in our experimental design and biochemistry, because we submitted the first paper and we said there is a certain preference of USP39 to recognize the mRNA molecules, because we saw that it was enriched in UA sequences. The problem there is that it was an artifact of in vitro studies. And we sent the paper, and one reviewer was expert in probably splicing, structural biophysics. This person knew everything. So he sent us 29 comments. In each of the comments, the person was right. So we did everything he suggested. We recalculated, planned, and we said that there is no specificity there. So that we knew. But we didn't know what is the consequence, what is the explanation of this. And then because we really wanted now to understand, so we connected to a very good group, this Stefan Legevi, who is a mathematical modeler of the splicing events. And he came out to calculate for us modeling about the stability of the complex. And he says there is no specificity, no sequence specificity. It is an issue that the splicing complex can jump left and right before and after the classical splicing event, and in that case you have an alternative jumping. And therefore in different cells we see the different splicing events. It's not the same genes. And of course with cells that have more splicing events like neurons, they have more of those compared to others. So there is a logic in this one. And then when our paper came out, the guy from Barcelona, Juan, calls me and says, "This is fantastic modeling. We in the field have been studying that for a long time. This all makes sense now." So not only that we got his confirmation that our data are correct, but we also got amazing credit by the person who is top in the field. So I think that's where we stayed at the moment, and we are still enjoying this type of freedom that the alternative splicing can be non-regulated.
Thank you for the great talk. I'm still not entirely clear why it is that USP39 is oncogenic. So is it because it prevents this underground splicing that creates proteotoxic stress? But at the same time, you said that tumors with high USP39 have high proteotoxic stress. So
Right.
how can you put that together?
Yeah, so I think that's a good, I asked several times question and I didn't answer it correctly, but it's true. It is true that the people have published that USP39 correlates with the higher aggressiveness and so on, and therefore they call it pro-oncogenic. When you see now, USP39 has nothing to do with oncogenic process. No. But it is highly correlative. More aggressive tumors, higher level of USP39. And that's where the description of pro-oncogene came. So now we describe this and we said, fine, this really correlates, and it is in this stage 3, stage 4, lung cancer, but not in all cancers. You have really high expression. And this high expression is part of the of this process. Yeah, so it's part of this process because the tumor cells have a high splicing demand. And during that time, USP39 grows very high. And it grows higher and higher. And this is probably not 100% compensating, but maybe 90. Therefore, you have still signature. You still have a signature of proteotoxic stress, but the tumor cells don't care about this. They boost it up. They boosted everything up, and they can live normally. They are not damaged because their autophagy and ubiquitin system operates to the extent that it's fully normal. So for us normal cells, that is enormous stress. For cancer cells, it's tolerated because it boosted all the machineries. And USP39 is one of the maybe Achilles tendon of this boosted system. Now, if you would just plug that out, things would collapse. And all of this stuff here collapses because it cannot tolerate it anymore and becomes prototoxic, much higher, much faster than in normal situations. So that's where we see connection between correlation in pro-oncogenesis and the weak spot when you get used to it, like addicted. If you would plug it out, you would kill the cells. And that's what we see in in vivo in mice.
That was very nice. Thank you. So I have a very naive question. Autophagy, so we know that metabolism is dysregulated in cancer. We know that autophagy has this kind of double-edged sword in tumors as well. Does the proteasome have this similar dysregulation in cancer, and is it more similar to the autophagy double-edged sword with a change in how it can be manipulated in tumors at different stages? And how does this relate to using the proteins, which are dependent on the proteasome for their function?
Right. So the proteasome has been exploited even before PROTACs. There was this MG132. It was one of the first one experimental, but then we had also bortezomib. Bortezomib was a major inhibitor of the proteasome that has been used before PROTACs for treatment of multiple myeloma, because people have observed that in multiple myeloma, proteasome function is limited and was specific for that type of tumor, not in many other solid tumors. So that tells you already the differences in the requirements for the proteasome. Proteasome doesn't have a different changes besides having a different type of proteasome, immunoproteasome, different subunits, and there one can find a different capacity in cleaving different peptides and producing different antigens and so on. So but there is no correlation of like proteasome depends on metabolism, and then also for the cancer, and then when you remove it, it's gonna impact on the cancer, like it happens in autophagy, because autophagy is much broader and much more connected to the source of all metabolites that we need in our cells. So it's a big difference, and probably that's the reason why it is easier to deal with proteasome, because it's more single unit than autophagy, which is all over in every cell and very complicated relation between metabolism, starving, stress, and the physiology. Touching this microphone too much is it's dangerous, right?
So when you inhibit USB39, you generate prototoxic stress, but you also generate a lot of neoantigens.
Right.
So what part of the response to your cortex do you think is actually a consequence of the immune system killing the tumor? So if you would do the experiments that you do with the hydrodynamic injection with immune deficient mice, would you see the same thing?
Yeah, I don't know the answer to this one, but we made these HUH cells in immunocompetent cells, in mice. So I think this, and the speed, what happens here, I would not say that there is a major contribution of neoantigens, and for that reason. Whether the immune system operates and works, particularly when the cells are damaged, I believe yes. So I think if we would remove the immune component, there would be a weaker response, but there would still be a response independent of, because the cells would be just killed. But there would not be an additional component of immune system. I think maybe Benoit can comment more on this, because that's immunology question. Within the time window of 4 weeks, I don't really know. This is not something, maybe I need to ask in China there, they will do that as well. I have to say the quality of the communication and the quality of data is impeccable. 20 years ago, we were normally buying the mice, knockout, knock-in, in America, paying $50,000, $60,000. Now, we ordered the mice for the Spartan conditional knock-in, 2 mice for 18,000 euros. 2. And I mean, the communication and quality of all the documents we received, we will hopefully get mice soon. It tells that these people know what they do. So and the prices are, I mean, who can do today with 20,000 anything with making, even if your own facility, it's very difficult to compete. So this tells us where we are in competitive ratios. This is not depressing note. We can still order it there, it's very good.
One last question before we end this. Maybe I have one. I'm not sure I understood the connection between lactate and the amount of USP39.
I also don't understand completely, but I think that Christian and Silla in the lab have shown that, I don't have a data here, but maybe if I go down, maybe there is something hidden here. Yeah, maybe it's here. I need to unhide and show it here. So, so this is what they were showing. They were showing that when you have a USP39 is triggered on by when you have a lactate added, you trigger on expression of USP39. And when you treat the cells with with this inhibitor of lactate, the USP39 level goes down. And they claim that this is linked, it's not shown here, but it's linked via MYC, because MYC is also triggered by lactate. High level of lactate activate MYC. And MYC has a binding site in front of USP39. So they connected it with lactate, MYC transcription, high level of USP39. So if you treat it all the time, you decrease even the normal endogenous level of USP39. So that is one arm of lactate, or oxalate, with blocking the lactate production. But I'm not so sure that this is the only one. That's why I was trying to be very careful there. But it was an indirect way that already tells us on the same way this is also happening, and we would see the phenotype.